before we get started, let's go over some basic vocabulary. Actin, myosin, the myosin head, troponin, tropomyosin, calcium ions, ATP, neuromuscular junction, sarcoplasmic reticulum, T-tubules, vesicles, and acetylcholine. Skeletal muscles will contract or relax when they receive electrical signals from the action potentials through the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. The action potential causes the vesicles to release the neurotransmitter acetylcholine that will then go to the synaptic cleft and bind with the ion channel receptors on the sarcolemma. The binding action of acetylcholine neurotransmitter to the receptor causes the receptors to open, allowing sodium ions, highly concentrated on the outside of the myofibril, to flow inside, depolarizing the sarcolemma. As sodium ions enter, voltage-gated channels open, regenerating another action potential. The sarcolemma will then convey the electrical signal along its length, where it will then be able to propagate down the T-tubule. This triggers the voltage-sensitive proteins that are linked to the calcium channels in the cell's sarcoplasmic reticulum. This then causes the calcium to rush into the rest of the cell.
This causes tropamine to bind very easily with calcium, which causes tropomyosin to also be pulled away from the sites on the actin strands that the myosin heads want to bind with. However, only myosin heads that already have an ATP molecule will be able to bind with the sites on the actin strands. However, the myosin heads then move into an extended position, holding the energy released with ATP broke down into ADP and a leftover phosphate. Myosin then releases all the stored energy and extends to bind with actin. This causes the myosin to change shape and pull an actin strand like pulling a rope. This causes the whole sarcomere to shrink and contracts the muscle. This part shows the sliding part of the sliding filament theory. The ADP and leftover phosphate then unbinds with the myosin head, which allows for another ATP molecule to bind to the myosin head. This new binding of ATP causes the myosin head to release from the actin. But the myosin breaks down the new molecule of ATP into ADP and phosphate, again causing the myosin to get into an extended position yet again. The myosin then binds again with the actin. The calcium pumps are still pumping the calcium into the sarcoplasmic reticulum, so they start grabbing the calcium that has bonded with tropamine, and the calcium from tropamine is released. This then causes the tropomyosin to get back in its shape and prevent myosin from binding with actin again.
This sliding filament theory can be applied to explain rigor mortis, which frequently occurs when an animal dies. The body's joints become stiff and unmovable. This occurs because the sarcoplasmic reticulum becomes permeable, causing the calcium ions to leave the sarcoplasm. Some calcium ions, however, still bind to the troponin, exposing the actin to the myosin. The myosin and actin bind, causing the muscle to contract. Since the body no longer breathes, ATP is no longer produced in the body. Since ATP is no longer produced, the myosin heads and actin will stay bound together because ATP won't cause the myosin heads to release. As a result of all this, the muscles stay completely stiff after the animal is dead.